Hi, I'm Sadashiva Pai from Science Mission. So we have today Dr. Bina Joe from University of Toledo with us. She is a distinguished university professor and chair, founding executive director, Center for Hypertension and Precision Medicine. She is also a principal investigator, programs in physiological genomics. Dr. Joe has won several awards to her credit. I'm going to mention only few of them here because otherwise it will take almost an hour to go through all of them. She's won President's Award for Outstanding Scholar in the University of Toledo in 2018. She is a Distinguished University Professor 2017. She has won President's Award for Excellence in Grantsmanship, University of Toledo 2017. She also won the University of Toledo College of Medicine and Life Sciences Dean's Award for Research Excellence in Sustainable Research in 2015. Several more of them. So welcome, Dr. Joe, to Science Hangout.
Hi, nice to be talking on this Hangout because this is a new thing for me. So everybody out there, hi.
Okay, can you please talk about yourself, your background, education, etc.?
I am basically from India, South India, from Mysore, a small town south of Bangalore. I did my complete education from kindergarten to PhD in that town. And then I moved to the Indian Institute of Science for my postdoc for a brief period, and then came to the NIH for my postdoc. And at the turn of the century, I moved to Toledo.
Okay.
Since for 18 years now. So my career path has been interesting. I worked on autoimmune diseases for a while, and then latched on to genetics at the NIH and moved that aspect of it to Toledo. I've been working on several complex traits. Hypertension is prominent for what we are funded for, and so I've stuck with this for a while now.
Okay. And let's talk about your mentors or people who supported you during your career.
Dr. Pai, as you know, there are a lot of people who serve as mentors, or you grab them for their roles for what they do. So my dual roles as scientist and administrator have seek out for different mentors for the science part of it. A few people, including my graduate school mentor, Dr. Lokesh from India, and Dr. Rapp from John Rapp Toledo, have been exceptional mentors. There are others that I can name for the administrative aspects. It's Dr. Cooper, who's the dean of our school. He's excellent. I do look up to women as mentors, internationally acclaimed folks like Anna Dominic, Brian Toyus, to mention a few. And my best critique is my husband, who is Venki Basarur from the University of Michigan. So that's a unpaid critique that I get for all we do. It's been exceptional. It's been a wonderful journey.
Okay. And other than research, I know you are busy with the research most of the times. Other than research, how else you spend time? What are your hobbies and all?
Well, I'm a trained Indian classical dance professional. So I do Bharatanatyam, which is what we do over the weekends. In fact, I have a picture for you to show in my presentation. I take pride in getting girls, mainly girls, excited for women in STEM through art. So I do a little research with the art part of it too.
Okay, that's good. And how big is your lab? Can you talk about your projects and lab people and all?
So lab people are the pudding that I get to eat for free. They are an exceptional group of people. There are about 7 graduate students. Some graduated out, some are in the lab, MD, PhD students including. There are a few postdocs, faculty, a big range of trainees who basically run the show for science. At this age, I think I'm receptive to their creativity more than banking on my own. So if my students are listening in, they'll know what I'm talking about. It's pretty obvious that their brains more.
Okay. And talk about some of the projects you are doing, and then we can go to your presentation.
So why not we switch to a presentation, because that kind of has slides in it as a so we won't waste time doing that now. Is that okay?
Okay, sure. Go ahead.
All right. So let's go on to presentation. PowerPoint, and that's not the slide I want to be on. Okay, are you seeing the slides now?
Yeah, salt, silver, ketones, hypertension. That's the one.
Perfect. Again, I will try and make it concise. If it's not, you can hurry me up as you please.
Okay.
Together we have 45 minutes. Okay. So the topic I put out for today is salt, liver, ketones, and hypertension. What are the links? The specific topic pertains to the article that we published recently. But before getting into it, in terms of addressing how our lab is asking different questions, I'll go through some of those initial work and transition into this topic. Right? So first off, this is the picture I was referring to. Of my students, they brighten my life. Here's my daughter, Megan Basroor. Kind of do it for her, but all of these are my people that keep my artistic sense alive and bring joy to coming to work on Mondays. This is what I come to work on for Monday through Friday, an exceptional group of individuals. Several are graduate students and postdocs. So I will be highlighting 3 papers, one from Blair as the first author, the second one from Sarah as the first author, and the third from Saroj as the first author. And the rest are part of the team for all the papers.
Okay.
Of course, work. Yeah, go on.
Can you please hide that sharing stuff? Like there's that hide thing. Yeah, so that it doesn't block. Yeah.
Sorry. It's okay now?
Yeah, go ahead.
Right. So over the years, we have had a number of collaborators. I wish to highlight Dr. Vijay Kumar, who's now with us. He came to us recently from Penn State. He's now the director of the Microbiome Consortium, with whom we work very closely on the projects I'm going to describe today. For the latter part, the microbiome part. So I had to give a shout out to him and his team for the exceptional work they do for us. So this is the paper, which I'm very happy that you showcased for us, Dr. Pai, in Science, a recent article on salt-responsive metabolite beta-hydroxy butyrate attenuates hypertension. We'll talk about it as the third paper. So let me go back to our roots. We do work with blood pressure, and you may or may not know that there's a new guideline based on a clinical trial. So anybody under 120 is normal. Above that number in the US is considered some stage of elevated blood pressure. So that has significantly raised the number for percentage of people who are hypertensive. For example, in Ohio, that takes it up to 64% of adults in Ohio are hypertensive. But from our viewpoint, hypertension is a risk factor that has a number of components. One is genetics, environment, and then everything else that you can bin in one group. We have been focused on the genetics. Evidence-based medicine suggests everybody is equal in genetics. That's not true. In clinical realities, we are all very different. And so the term precision medicine is kind of attractive to go towards future of medicine, as you know. And we model it. We model it in animals for how they would behave with different genes for their presentation of blood pressure. The animal model that we use in the lab prominently is called the Dahl salt-sensitive hypertensive rats. These were inbred in Toledo. We do study not GWAS, which is association studies. We study genome-wide inheritance, which is familial transmission of genes through this model for what causes hypertension from the genetic viewpoint. We have used recent tools to sequence the genome. We know our finite set is a cluster of 2 million base pairs that's different between the hypertensive rat and the resistant rat. So this being the case, which ones are responsible for blood pressure control in this animal is what we are after. The models originated in Brookhaven National Laboratory. Louis Dahl was the one who created the S rats. He was keeping outbred populations alive, and we brought it over to Toledo and inbred it. My predecessor, John Rapp, did it. So the history of it is salt has always been a topic associated with this salt-sensitive model, but it always comes up in new forms, which is what we'll discuss in our third paper today. This is more of about Dr. Dahl. It's a fascinating history for how during World War II, science was still alive in minds of curious people. So if you want to go ahead and read this, you'll get a personal history of Dr. Dahl and how the work has come about using this model. Using this model, I'm going to, go through a few decades of work. We have published a lot trying to identify genes, positional clone them. Some of these papers are highlighted here. This is, again, our history. One of the most recent papers that I'm very proud of is from my graduate student, Shi Cheng, who's now a postdoc with us. We CRISPR engineered 19 base pairs on this rat and identified that those are positionally cloned nucleotides contributing to blood pressure regulation. When we came to this point, the question was, what's translational? How do we go? So it's a difficult world, as you know, in genetics. So even though we can take it to final proof in models, which was what the question we asked initially, there's still a lot more work to do in genetics. We have reviewed all of this, and I'll highlight this review for you. It's pretty substantial for both human hypertension and models, if anybody's interested to look at the genetic aspects of what we do. So this question of which genome stared at us around 2014, call it kind of a midlife crisis in the lab. You're looking at the mammalian genome, you're looking at a rat genome, trying to simulate what goes on in the human genome. But then we are thrown with this idea that there is another big genome, collectively called the microbiome, with way more genes that's actually impacting traits compared to human genome, even more so. So do we call the microbiome as a genetic aspect? Well, you can question that. If you refer to the biome, it's the genes, it's genetic. If you refer to the biota, it's more physiology. So it's too complex to go into the biome, so we started looking at the biota as a question. As you know, microbiota alter almost every facet of human life and animal life, so there's a lot to study there. I don't have to harp on this at all, except to point out that when you look at PubMed, which we published this chart not too long ago, this is a graph of number of publications versus years for a search term microbiota in PubMed. You will see it picked up around the turn of the century because of sequencing capabilities and has sharply risen for all diseases. Now, when you subset it out for just microbiota and hypertension, you'll see how tiny the number of publications are. It isn't updated, but I can tell you more than half are reviews. So there's not much original work in linking microbiota to blood pressure as a trait. So we asked that fundamental question and reported it first in 2015 that there is a link between gut microbiota and hypertension in the Dahl rat, the rat we studied. This was highlighted in 2015 by the AHA, but then not many worked on it since, so the numbers remain small for publications. However, it's catching on. Having made that fundamental discovery, I just want to briefly report what that paper meant for us. The S and the R rats, which are the Dahl salt-sensitive rats and the salt-resistant non-hypertensive rats, their microbiotal compositions are different. That was the first observation. So then the question became, if we swap these microbiota to these animals, will their blood pressure change? We did that experiment. We swapped gut microbiota from a resistant rat to the S rat and had an autologous control. Vice versa, S to R and an autologous control. And the cecal transplant experiment gave us this data. So to the salt-resistant rats, regardless of what microbiota you gave, their blood pressure remained unchanged. Systolic blood pressure plotted here went up highest for the R to S group. compared to the S to S group. So pay attention to this. R is a normotensive rat, the microbiota of which, when we transferred to the susceptible hypertensive rat, the blood pressure of this rat further increased. So this gave us an indication that there is some sort of symbiotic relationship between the gut microbiota and these animals that we needed to study. It wasn't as if the normotensive rats had protective microbiota. It had to do with which rat it was exposed to for effects on blood pressure. Anyway, we finished up that paper doing short-chain fatty acid analysis, which are metabolites of microbiota, as you know. We found that in the animals that had the raised blood pressure, acetate was higher, and so was HEPTANOATE. And these were early years, so we couldn't go any further than this. And we finished up that work with the conclusion that there is a definitive link. We just need to mine the data more on what it is. The concept also evolved for us that some of the groups were looking at gut-renal axis and gut-bone marrow axis, and we came out with the holobiont concept to be front and center for, instead of looking at these individual axes for how microbiota influence blood pressure, we perhaps need to look at the whole organism and identify newer ways of how microbiota are impacting to change normal blood pressure to hypertension. The second study what we did, therefore, was with antibiotics. As you know, antibiotics are bactericidal, and so is salt. And salt increases blood pressure. So what about antibiotics? The simple logic, if it is through microbiota, which there was no data for at that time, then antibiotics should also have some impact on blood pressure, which was scarier thought because of how often we use antibiotics in our lives. But that's the question we asked to see what that means to hypertensives. So we took 2 strains of rats. I'm introducing the second strain here. First one is the S rat, which is genetically hypertensive. Second is the spontaneously hypertensive rat, which was selected without any salt diet, but is still hypertensive. So we took 2 models and asked the question, when we treat them with antibiotics, what happens to their microbiota? Here you see the red lines depict the alpha diversity, which is all lowered in the antibiotic-treated groups. We had 3 antibiotics, minocycline, neomycin, and vancomycin. And as expected, their microbiota clusters were different, indicating that the antibiotics were performing their job of clearing gut microbiota selectively so for what they target. But what was interesting was that in some of the rats, in the same rat strain, the 3 antibiotics did not work identical. For example, in the S rat, you will see that the red line is the control. Blood pressure of all the 3 increased, but in the SHR, there was no effect of neomycin, but the other 2 antibiotics decreased blood pressure. So strain-specific effects of antibiotics, in one it increases, in the other it has disparate effects. So try to look at this graph and think about us, individuals. We are different for our antibiotic sensitivities because we are genetically wired differently for our susceptibility to blood pressure, which means we don't know if we are hurting ourselves by taking antibiotics or helping ourselves by taking some antibiotics. This is actual personalized medicine for me in action, demonstrated through models. So this was an important study for us to point out that antibiotics need caution for prescription by doctors for hypertensive subjects who may have different genetic propensities. Again, this is early times, so we don't know what this means in terms of mechanism. So the third study I'm going to describe now, we actually thought of hitting mechanism a little harder than what we had done with the previous 2 studies. So in this study, we asked the question, does salt influence blood pressure via the gut microbiome? So this is a nice diagram from a Nature Reviews Cardiology article. where they did mention the human genetic aspect, which I talked to you about for what we were doing, and we are still continuing to do. But then we brought in the salt as an environmental factor, dietary factor, and asked the question, well, is the genetic link of microbiota to the genome impacted by an environmental agent? And so we said, let's study salt. So we looked at the salt effects on our models. First off, we fed rats a low salt diet or a high salt diet. The chart here in black is the systolic blood pressure of an animal, of the group of animals that were on low salt. And this red line is that of high salt. As you can see, this is not new data. When we eat salt, blood pressure goes up. And this is very high in this model. So this is the model system. In these animals, when we compared their microbiota, night and day different. So clusters, PCOA plots, indicate that there are big differences when animals are consuming high salt or low salt in their gut microbiota. So then we looked at certain classes, which ones went up and down. Lactobacilli, shown here, were lower in the high salt group compared to the low salt group. This was not new data. At the time we were studying this, there was another group in Germany that did the exact studies and published in Nature, Dr. Muller's group, that this is what they observed as well. So it was somewhat, corroborative at that point. What we also noticed was that in the high salt group, proteobacteria are lower and Prevotella is higher. So with these three, there were other changes, but we kind of were looking at a spread of what is altering, and these struck us for changes. The next question we asked was, because microbiota alter metabolomes, let's go ahead and check the plasma of these rats for what are the metabolites circulating in these animals and how different are they. So we did a carbohydrate-specific panel for, honestly, a serendipitous reason. That was just the first one. We said, okay, let's go ahead and look at that. What my graduate student did very clever, Saroj, was he fasted the animals and then studied them. And here you're looking at a volcano plot where you see the red dots are the significant metabolites that were different between the low and high salt fed groups of animals. We focused in on beta-hydroxy butyrate, which was lower in the high salt fed group. This was, I think, one of the highest differentially expressed metabolites. And because of the fact I told you it was fasted rats, it made a lot more sense to look at beta-hydroxy butyrate, as you know, is a metabolite that is altered in calorie restriction and fasting and exercise and all of that. But it had no known links to blood pressure regulation. So it was interesting for us to pick this up for further work. The first experiment we did was to confirm that this is all true. So we took the metabolomics data to heart and measured what the actual plasma circulating levels are. So here you're seeing beta-hydroxy butyrate in fasting and fed conditions of low and high salt shown in black and red. You will see that in the fasted state, we were able to, validate the observation through the metabolomics study. And it was not significant in the fed state. Now you can appreciate why the fasting state was key for us to get to this data. So the next question we asked is, well, if that's all true, it must be very simple for us to check if we exogenously add this ketone body into what's available in the rat's body, can we lower its blood pressure? We're asking a tall order question, but if there's a link, we should see some result here. So the way we approached this was to use this chemical called 1,3-butanediol, which is an FDA-approved drug for weight loss and glucose control. This is a precursor of beta-hydroxybutyrate. So we designed the study to feed the animals on a high salt diet with 1,3-butanediol to observe their blood pressure. Before we got the blood pressure data, we had to be sure that this compound does convert into beta-hydroxybutyrate. So after gavage, we did measure their beta-hydroxy butyrate levels, and you can see, as this is a very high concentration, it's 20% volume by volume gavage, both fasted and fed states, it is remarkably high, which is what we wanted to do. The question then is, this high level of BHB, does it then alter blood pressure? And indeed, when we gave the animals 1,3-butanediol, the line that you see in purple here is the systolic blood pressure of the group that received the 1,3-butanediol despite being on a high salt diet. The controls are just on high salt. And you'll appreciate it that there is a significant dip at every time point for systolic blood pressure. This is the money slide for there is something profound happening when you alter ketone body in a rat that lowers its blood pressure. Question is, what did we do so magically that it lowers its blood pressure? Okay, all of this information, we then ran to the patent office with it because this is pretty exciting as a potential therapy for salt-sensitive hypertensives. The next question was, circling it back to microbiota, did what we observed, was it due to the changes in microbiota or not? Well, I can tell you, we don't have an answer yet, but I can tell you how we're going about it. The oral supplementation of 1,3-butanediol also alters the gut microbiota. As you can see, purple group is one with the ketone body precursor, and the red one is just the high salt group. And these are the PCOA plots. Coming back to what the microbiotal status was, this was just the comparisons to reiterate to that this is the top panel is the difference we had with low and high salts that I had shown you previously. If you recall it, lactobacilli went down. Here, lactobacilli appeared to go up, but didn't make statistical significance. Proteobacteria went down, but when we were giving the ketone body, it seems like it is getting reversed. Prevotella went up in the low to high salt comparison, but when the high salt group was given butanediol, it's looking like it's reversed. So there are some reversals, but not the one that was in focus with the other group publishing in Nature, that lactobacilli in fact were altered by salt, and feeding lactobacilli rescues blood pressure. May not work in our model, A, and B, the study that we are conducting with ketone bodies in focus may or may not work with lactobacilli. For us, it looks like it may not work so much with lactobacilli, but it may be associated with changes in other microbiota. So that part of the study is still not complete, but we will be working on these aspects for knowing the links between microbiota and ketone bodies of the host. Regardless, we still had to answer how blood pressure was lowered in these animals given ketone body precursor. So first we checked the obvious spots to check, which are gut permeability or serum immunoreactivity to microbial products. None of them were different between the three groups that we were studying: low salt, high salt, or high salt plus 1,3-butanediol. So we concluded that alterations in gut permeability do not explain the blood pressure differences observed with 1,3-butanediol. Flagellin or anti-flagellin and anti-LPS levels, which are microbial products, anti-products formed by the host, are not altered. So it's not through bacterial reactivity that this blood pressure difference is explained. Then we looked at what I'm not showing here is the TH17 profiles, that they were also not different. I'm just skipping that data. Well, what we found, however, was a very nice correlation between NLRP3 levels. Now you're looking at high salt and high salt treated with 1,3-butanediol groups. NLRP3 in the renal tissue is lowered. CASP1 is lowered. And so are IL-18 and IL-1 beta. And here you're seeing the protein level, serum IL-1 beta. So all of these data indicated that there is a inactivation of the NLRP3 inflammasome in the presence of 1,3-butyrenediol. And that isn't new because BHB is known to inactivate NLRP3 inflammasome. What is new is that this is operational to control blood pressure is the new aspect of it. So then we went ahead and did some renal histology. Here you're seeing the renal injury score being lowered, the protein casts, excuse me, and fibrosis in blue are all significantly lower compared to the top panel, which is the high salt group. In the lower panel is the one treated with 1,3-butyrenediol. So there's a rescue of renal function, renal architecture, anatomy, and physiology. We also went ahead and looked for macrophage infiltration. As you can see, the brown stain here in the high salt group are macrophages infiltrating the renal tissue, and that's all lowered in the treated group. This is a quantitative score indicating that, well, the kidney is probably better, and therefore infiltration of macrophages is lower. And this lowering of improvement in kidney function is one of the ways by which blood pressure lowering could be impacted. So that's the paper we wrote, which a kidney in focus for cell reports. And the key points here are that the new aspects are the dietary salt, alters plasma beta-hydroxybutyrate, meaning it lowers it. And thereby we see a correlation for increased in blood pressure. We sought to correct the plasma levels by a nutritional intervention with a precursor of beta-hydroxybutyrate. And then we observed lowering of blood pressure, but then we followed the mechanism through the kidney. We are able to demonstrate that via inactivation of the NLRP3 inflammasome by blocking it, lowering all the downstream mediators, we are able to observe three key parameters: lowering of serum sodium-potassium ratio, lowering of proteinuria, and lowering of renal injury score, which are all known features for contributing to blood pressure. And therefore, we interpret that this rescue of renal function is what is lowering blood pressure mechanistically. This is just one article. BHB has influence on other tissues. So right now we are focusing on other tissues for what and how it could impact, for example, the brain, blood vessels, etc., for how that would also contribute to blood pressure regulation. The key part that I want you to focus on is the diagram here of the liver. From what we have read about hypertension, I don't think liver is a prominent organ that we study in blood pressure regulation. So here's an ounce of data to consider that liver may prominently contribute to blood pressure regulation. It may also link the aspects of metabolic syndrome wherein hypertension presents as a significant risk factor. So there are more studies to be conducted for how the liver may impact hypertension. So the take-home message from this third paper is shown in this cartoon. Here you have a nice balance between salt and BOHB, which is beta-hydroxybutyrate, normal blood pressure, which could be tipped in conditions of hypertension, which are due to elevated salt. And in that same condition, our observation was that beta-hydroxybutyrate is lowered. And our experiments have proved that by rescuing this back, we are able to control blood pressure, regain normalcy as best as we can state it, despite the usage of high salt by the animal in its diet. So when you relate this back to what happens in exercise, BOHB is higher, is elevated in exercise. So one of the ways by which exercise may help hypertensives despite their salt consumption could be because they elevate their beta-hydroxybutyrate levels. So what we are suggesting is Doesn't matter the means, if it's either through exercise or calorie restriction or fasting, all of these conditions increase BHB, or by nutritional intervention means like we suggest, to take a bolus of BOHB or its precursor, it may be a way to think about controlling salt-sensitive hypertension in humans. The other part to consider is if this is impacting many tissues, we may not have to look for selective agents that target exclusively the kidney or the blood vessel or the brain, or other organ-specific therapeutics that are in the market for blood pressure control. We may be looking at a global impact on several tissues in one go, which all benefit to keep blood pressure normal. So that would be one way of, igniting thinking for how the liver could impact blood pressure regulation. So bringing it all front and center, I think 2 more minutes. We started out with this slide that we were working on the genetics for each individual is different, and therefore the genetic compositions may have to be carefully evaluated for future doctors to look at their DNA blueprints and overlaid with environmental factors for preventing, treating, addressing hypertension in the clinic. However, a new factor is the microbiome, which is also highly variable between individuals. The metabolomes are also variable. Are there groups, subgroups that we can target with data on metabolomes and microbiome data in hypertensives? Well, that's a question to address. And in the lab, currently, we are slowly moving in that direction for addressing that exact question as shown here for what's the translational significance of knowing that certain metabolites are altered or certain microbiota are altered in individuals. We are currently recruiting subjects for a study that we have an IRB approval for already. What we are looking for are people with high blood pressure and people with normal blood pressure, to compare the two groups, to study the roles of their bacteria and metabolites circulating in their bodies, to see if there's any association for these two factors as we study in our models. So the phone number is here and the email contact is here for whoever is listening. If you could pay attention to how important it would be for us to collect these data, please reach out to us. We will be very grateful to you for doing so. With that, I think I'm done. I can take questions, if any.
Okay. Let me see. You can just get out of this presentation mode.
Sure.
Just double click. Yeah. I want to see if there are few people on this thing. If anybody is interested in asking any questions.
Okay. So let me. Am I out of this already now?
Not yet. It is just going, but I don't see you. You should be seen on the screen, basically.
Okay.
One thing you can do is there is on the left side screen share. Double click on that.
Okay.
Top. Yeah. Second. Yeah. Here you go. Okay. Thank you so much. It's pretty interesting, and I have glanced through that your cell reports paper recently, so which is pretty interesting. I have a few questions.
Yes.
If you don't mind. One is you looked at the metabolome, right?
Yes. Can you hear me?
So yeah, I can hear you.
Okay, great.
Can you?
Yes.
Okay. Looked at the metabolome, like since the bacteria are in the gut, have you looked at any urinary components or the fecal things which is changing or anything happening with that?
So the reporters for what's in the gut, we have used only fecal material. In some of our studies, we have used cecal samples. In others, we have used fecal samples. So that's
okay.
We have not looked at the urinary microbiome yet.
I'm not talking about the microbiome. I'm talking about the metabolites. Like when you add salt and all, there will be changes in the metabolite which might be secreted in the urine and fecus.
Right.
You found that beta-hydroxybutyrate from the serum, right? So I'm just thinking, like, is there anything coming out from the urine or in the fecal matter, something comes out, which could be other way to look at it.
So your viewpoint is very well taken. If I can fast forward that thought, can we use
Yeah.
pH levels in the urine or stool samples as biomarkers would be one way of looking at it for the extent of blood sugar. We have not done it for, well, the initial report we haven't. What we are doing right now is to titrate the amount of BHB that is really required for the observed physiological change in blood pressure. And in that context, we will have to monitor what you just mentioned in the urine,
Okay.
the other sample.
Okay. Okay. And the other question is like the fiber, right? Fiber plays a role in changes in the microbiota and all those things. So have you looked, because and fiber also, like some people say, it will decrease your blood pressure too, like if you add fiber to your food. So have you looked into that, or anybody else has looked into that in terms of looking at the blood pressure, microbiome, and fiber content?
So you're asking questions for other projects in the lab.
I'm just giving you some ideas.
Great ideas. We are looking at the effects of fiber. We have some data that I don't think is reasonable to air in this context because it's all preliminary.
That's fine. That's fine. Yeah.
And there's also dietary additives we are looking at. Some of them used in ancient Indian culinary, not ancient, even now, like curcumin and capsaicin. We're looking at the effects of those very carefully, but because we know they're all anti-inflammatory too, in addition to being antibiotic. Question, well, how do they then operate in the context of diet for influencing blood pressure? There are indications similar to fiber for some of these food additives to alter blood pressure, but there's no rationale, like big evidence from the microbiotal frontier. So that's an area we're working with. So appreciate your question on the fiber aspect.
Okay. And the last one would be, like you talked about the brain, right? Like what happens, beta-hydroxy butyrate and all those things. And there are recent reports in, I think in Science or Nature somewhere or Cell, like there is direct connection between the brain, I mean like the gastric intestinal nerve endings with the brain. So it directly communicates with the brain. And certainly brain plays a role in the hypertension and all those things, modulating the adrenal glands and all those stuff. So are you looking in that direction too? Or like, because since it is microbiota, it's I think it is, like I'm just thinking it, I mean thinking in terms of openly about the whole thing.
Dr. Pai, you're spot on with that question because beta-hydroxybutyrate is known to, I mean, it is an alternate fuel for the brain. So there are reports that BHP consumption keeps the brain alive more so. I'm saying it in layman terms. What we are observing is that our animals on 1,3-butane-diol are active. So we are looking into the brain functionality part of it to see how cognition could be affected. And you know, the brain has a huge impact on blood pressure regulation. So is that I was alluding to other tissues, brain is also in it for how that might impact blood pressure through the alteration in ketone bodies. I want to circle back on the fiber question just for a minute, if I may.
Yeah, sure. Go ahead.
Yeah, Dr. Vijay Kumar has recently published
a Cell paper, right?
Yes, you're on that as collaborators. What we are looking at is the fiber aspect of it. So the specific animal model that was presented there is the toll receptor 5 knockout mouse. The initial study from his previous work in Science demonstrates that TLR5 knockouts have elevated hypertension. But when the same animals are given soluble fiber, they have effects on liver cancer. So soluble fiber, if you put it into context of blood pressure regulation, there is an aspect of it that we may be able to work with the TOL5 receptor knockout mice. I would recommend a hangout with Dr. Kumar's lab to understand.
I'm thinking of, he's in my list.
Yeah, it will be a nice way of getting from here to the next step. Yeah.
Put in a word for me. I'm going to contact him within next, because he is in another 8-10 people I'm supposed to contact for this hangout.
Oh, sure. He's in our department. I'd be glad to do that.
Yeah, yeah. I'll send him an email to in next few days. But if you could put in a word, that also. And one more, the last one is like ketone bodies are like, you have when you eat a lot of fatty food, like it's a fat, right? When you digest fat, there is a lot of ketone bodies.
Mhm.
So how does it like relate? Like if you are a obese person, you have a higher amount of like people say if you are higher, like your hypertension goes up, but there would be more of ketone bodies. I would assume. I don't know. I haven't looked into literature or anything like that. I would assume there would be more ketone bodies in there. How does it correlate? More deeper needs to be looked into there. But just I'm curious to figure out like if you have thought about that. Certainly you would have.
Certainly it's not true because your viewpoints are coming in as important for us to consider. When you're mentioning
Okay.
subjects, their ketone body levels are higher or lower? Is what you,
Higher.
yeah,
I would say because there's more fat. For fat metabolism would be higher compared to see normal people because
so
of
that
the higher fat content.
yes, so that is new information for me per se. We'll have to look into
Mm.
that. There are two
Yeah.
ways of looking at it from a hypertension researcher point of view. Not all obese people are hypertensive.
Mm-hmm.
Not all
Yeah.
lean
That's true.
people are normotensive.
Normal tension. Yeah, that's true.
There's disconnect there and perhaps connection in some subset. So one
Yeah.
way to design experiments for testing what you said would be to feed high-fat diets to understand ketone body regulation in a high-fat diet context,
Definitely.
high
Yep.
salt.
Yep.
And
Yep.
models have to be carefully thought about for which ones to
Yeah.
study.
Yeah. 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 I think I don't have any more questions. So I really appreciate you taking time to do this. I'm sure you are busy. Thank you so much. And if anything comes up in next few months or a year, give me a call so we can do another hangout. Thank
Sure.
you so much.
Dr. Pai, I have a question. Are there people that can ask
Yeah.
questions on this hangout or no?
No, it's basically there are few people, 3-4 people are viewing it. What happens is like it will be on the website once it is done. Once I stop it, automatically it goes to our website. Anybody can watch it, and they can like there is a window there. Like if they want, they have any questions, they can ask questions
All
basically.
right.
So
Great.
that's the like they can directly in touch with you basically.
Well, thank you so much for this opportunity. I've had fun doing this. And hopefully, if there are questions, we'll be happy to answer them.
Yeah, certainly. Thank you so much.
We'll keep in touch.
Bye. Yeah, sure.